Hello everyone, welcome to today's Revive webinar by the Global Antibiotic Research and Development Partnership, GARDP. My name is Victor Kwasi, and I'll be hosting today's session on repurposing drugs to address the crisis of antimicrobial resistance. Revive is GARDP's education and outreach program. It aims to connect and support the antimicrobial R&D community by facilitating learning, sharing knowledge, and connecting people. These webinars are part of our educational activities. All webinars are recorded and are freely available to view after the live broadcast on our website revive.gardp.org/webinars. You can also read our series of articles known as Antimicrobial Viewpoints, where experts discuss various topics within the field. Our latest is a piece written by Angela Hüttner from Geneva University Hospitals, as you can see here. We also have a resources section that includes the Antimicrobial Encyclopedia. This has definitions on various terms in the field, and many of which you'll hear in today's webinar. Some of these also include videos where experts give a further explanation on the terms. As always, today's presentations will be followed by a Q&A session. You can submit your questions at any time during the webinar by clicking on the questions button, as shown here. We will address the questions after the presentations and do our best to respond to as many as possible. If your question is for a specific speaker, please do indicate their name at the beginning of your question. Today's speakers are Freda Jen, Anthony Coates, and François Franceschi. Our moderator today is Jennifer Smart. Jennifer Smart is a senior microbiology consultant for the Serious Bacterial Infections Group at GARDP. She has experience in managing non-clinical development, clinical trials, and providing scientific and medical affairs support for anti-infectives. She received her PhD in microbiology and immunology at the University of Maryland in the USA, and went on to undertake postdoctoral fellowships at the University of Maryland and the University of Illinois in Chicago. Jennifer has 15 years of anti-infective drug development experience for both antibacterials and antifungals, and has previously worked at pharma companies including Basilea, Therapeutics, and Astellas. Welcome, Jennifer. I now hand over to you to start today's webinar with a brief presentation.
Hello, everyone, and welcome. Thank you for joining us today at Revive's webinar, Repurposing Drugs to Address the Crisis of Antimicrobial Resistance. As Victor said, my name is Jennifer Smart, and I'm a microbiology consultant with GARDP, and I'm delighted to be your host and moderator for today's session. So antimicrobial resistance, or AMR, is one of the most urgent threats to global health today, contributing to over 5 million deaths each year. And without urgent action, AMR-related deaths are projected to rise by 70% by 2050. So next slide. Despite this growing crisis, our antibiotic development pipeline remains under significant strain. Only a small number of new antibiotics are in development, or even fewer offer novel mechanisms of action to overcome resistance. Developing new antibiotics is complex, costly, and time-consuming, and often takes more than a decade to bring a new drug to patients. Even though the right antibiotics may not reach the regions most burdened by multi-drug resistant infections due to hurdles such as cost and lack of access to these new antibiotics. Next slide, please. Given these challenges, repurposing existing drugs, that is identifying new antimicrobial uses for approved or investigational medicines, has emerged as a promising and practical strategy. This offers us a way to reduce development timelines and helps preserve the effectiveness of current treatments. So today we'll explore how repurposed drugs can play a key role in addressing AMR, and I'm looking forward to the 3 expert presentations we have for you today. So our first speaker is Freda Jen. We can go to the next slide. Thank you. Freda Jen is a molecular microbiologist with a PhD focused on pathogenic Neisseria. Her research focuses on host pathogen interactions and molecular mechanisms that drive bacterial virulence, with the ultimate goal of developing new therapeutics and vaccines. Building on her expertise working on bacterial protein post-translational modifications, she contributed to the development of 2 gonococcal vaccine candidates, which are expected to enter human clinical trials soon. Her work also focuses on repurposing existing drugs and designing new antibiotics to combat multi-drug resistant Neisseria gonorrhoeae and other antimicrobial resistant gram-negative bacteria. I'm now happy to welcome Freda. Freda, the floor is yours.
Thank you, Jennifer. Next slide, please. I don't know. Awesome. Thank you. Hi, everyone. My name is Freda. I'm from Institute for Biomedicine and Glycomics at Griffith University in Australia. And first of all, I would like to thank GARDP for the invitation to speak today. And I would like to present our work on repurposing drug to address the increasing crisis of antimicrobial resistance, particularly in Neisseria gonorrhoeae. Sorry, this is not going to the next slide. Wonderful. Okay. Neisseria gonorrhoeae was first discovered by Albert Louis Sigmund Neisseria in 1879, and that's called Neisseria. Sorry, because I'm in Australia, it takes a while. It causes a sexually transmitted infection, gonorrhea. And gonorrhea is currently the second most commonly reported STI worldwide. And for the gonorrhea, the common sites for gonococcus infection include urethra, cervix, rectum, pharynx, and the conjunctiva. You probably will be surprised. Most of the gonococcus infection are asymptomatic, especially in women. Symptomatic infection can include urethritis in men, cervicitis in women, and the pharyngeal soreness, or it could be just as minor as like a burning throat. However, untreated or undetected infection can lead to PID, infertility, neonatal blindness, and also associated with the increased HIV transmission. And what about the treatments for Neisseria gonorrhoeae? As you can see the poster here, this is a poster during the time in 1944 attached to a curbside. Sorry, this is a rubbish bin, not a mailbox. And this is to spread the word to the World War II soldier that a single tablet of a penicillin was able to kill gonorrhea just in 4 hours. However, what about now? And as you can see the figure here, this was published by Agnes Newmore and Bill Sheffer in 2014. And as you can see here, from penicillin till now, Neisseria gonorrhoeae has developed resistance to most of the antibiotic used. And at that time, they predicted that after 2020, the superbugs, which is untreated gonorrhea, will become reality. And this was published in 2023. I really like this figure because this figure illustrates and shows how Neisseria gonorrhea develop resistance and then the resistant mechanisms to all the antibiotics used. And these are all the facts that global cases of Neisseria gonorrhea are rising. XDR and MDR strain challenge currently treatments and resistant to ceftriaxone, azithromycin, and also others are increasing based on the WHO and the CDC data. And treatment failure have been reported in Europe and the Southeast Asia. And most importantly, there's no vaccine available. And we've been addressing this issue by studying repurposing drug PPT2. And what is PPT2? PPT2 is a zinc ionophore, although it also interacts with copper. It was designed to target zinc and copper dysregulation to reduce brain toxicity as a potential treatment for Alzheimer's disease and Huntington disease. It passed the phase 1 but discontinued after phase 2 human trial because due to its ineffectiveness. And in the clinical trial, an oral dose of PBT2, 250 milligrams per day, was safe and well tolerated in Alzheimer's patients. PBT2's research and study was originated in the Mark Walker's group in the University of Queensland. And what they have found is in 2020, PBT2 combined with the zinc can synergize with the antibiotics, current antibiotic, to reverse antimicrobial resistance there was in a whole bunch of gram-negative bacteria, particularly in E. coli, Klebsiella, Acinetobacter, and Pseudomonas. And in the same years, we were also working on PBT2 with zinc can also synergize with the polymyxin B and the colistin to reverse the antimicrobial resistance to the antibiotics. And here's the data from the paper, and we show that these are the whole bunch of Neisseria gonorrhoeae we tested. They're all highly resistant to the polymyxin, highly resistant to colistin and polymyxin B. However, in the presence of PBT2, they become risk sensitive to colistin and the polymyxin B. To investigate the mode of action, we know that Neisseria gonorrhoeae are highly resistant to polymyxin B and also colistin, all the cationic peptide, due to the presence of this modification called phosphoethanolamine on its lipid A, which because of the charge, it can repel the negative charge cationic peptide. And the LPTA, you can see here, is an enzyme that's responsible for adding the phosphoethanolamine to the lipid A. And we therefore examine the lipid A structure of the wild-type strands grown in the PBT2 and zinc, and compared to the control wild-type without the PBT2 and zinc, and then also, of course, the LPTA mutants. And as you can see the result here, wild-type in PBT2 and zinc have a reductions of phosphoethanolamines lipid A compared with the wild-type. And we know that the enzyme LPTA is a metal cofactors enzyme, which contains 2 zinc and 1 magnesium. And PBT2 zinc, this is what we think, PBT2 and zinc may disrupt the LPTA function, and perhaps by impacting its metal cofactor and thereby reducing the activity, causing reduced transfer of phosphoethanolamine to the lipid A. And here's the summary slide showing that PBT2 and zinc can synergize with the polymyxin B and colistin, and that can reduce the potential modification of this phosphoethanolamine on the lipid A, which allowed the polymyxin B and colistins enter into the bacteria cell and then kill the bacteria. And because we know that there are 2 nature cationic antibiotic peptides available, so the following year we published that PBT2 and zinc can also synergize with these 2 nature cationic antibiotic peptides, and then to reverse antimicrobial resistance to this peptide. In 2022, we started working on looking at if we can have a PBT2 alone to be a new treatment for Neisseria gonorrhoeae. So in this study, we start with knowing that the PBT2-MIC against the WHOZ, the single-strand multi-drug resistant Neisseria gonorrhoeae, is 0.313 microgram per ml. And this is actually 10 to 100 times lower than what has been published in other bacteria. And we also did do the reading of a phase 2 clinical trial. And in the study, they showed that the plasma concentration of a PBT2 for the patient receiving 250 milligram per day for 72 hours, is about 1.6 microgram per ml. And that is about 5-fold higher than its MIC against multi-drug resistant Neisseria gonorrhoeae. This gives us an idea that for the patient to have those, the safety dose will probably enough to kill Neisseria gonorrhoeae in the body. And we next tested all other Neisseria gonorrhoeae strains, including the ATC strain, the laboratory strain MSC11, and also 13 multi-drug clinical isolate strains. And then they all show very low MIC value of a PBT2. So here we know that Neisseria gonorrhoeae particularly is exclusively sensitive to PBT2. So to further analyze the antimicrobial activities of PBT2, a time kill assay was performed. And as you can see, the data here show that PBT2 at the MIC 0.313 can kill Neisseria gonorrhoeae within 3 hours, indicating its bactericidal activity. And our major issue with antibiotic used to treat Neisseria gonorrhoeae is this pathogen's ability to rapidly develop resistance to the antibiotic use. And to address this, we performed the 30 consecutive daily cycle of the subculturing with the sub-MIC PBT2 treatment. And our data show that, as indicated in the orange dot here, no resistance emerged after 30 days of exposure. And humans are the only known reservoir of Neisseria gonorrhoeae. To evaluate the efficacy of PBT2 in the most biological relevant system, we use an established ex vivo human cervical epithelial cells, PECs, gonococcus infection, which was established by our collaborators, Jennifer Edward. And as shown here, PBT2 at the concentration, I'm very sorry about the typo here, but if you look at here, at the concentration of 0.016 microgram per ml, it was able to eliminate closely to 92% of Neisseria gonorrhoeae. And taken together, our data suggests that PBT2 holds a strong potential as the novel treatment for Neisseria gonorrhoeae. And next, given the promising efficacy of a PBT2, we next want to understand how Neisseria gonorrhoeae responds to PBT2 at the molecular level. And we perform RNA-seq to examine changes in the gene expression following the treatment. And as you can see in this result, orange dots indicates the gene expression following, the downregulate gene encoding NADH associated protein, while the blue dots here are the genes that involve in metal homeostasis. And notably, I would like to mention these genes, MPER. This is an iron repressed genes, and we found it's upregulated in the PBT2, in the presence of PBT2, And why is it so important with this is because when this is upregulated, it can downregulate the MTRF gene, which is MTRF is a pump, is a membrane pump, and decrease expression of MTRF means that will increase the sensitivity to detergent. And we did this, confirm this by experiment. We showed that in the presence of PBT2, the bacteria will increase the sensitivity to Triton X100. We next then want to know that there is another pathogenic Neisseria, which is Neisseria meningitidis. If these strains are also sensitive to PBT2. So we performed the MIC against 8 different serogroup of Neisseria meningitidis. And as you can see the data here, all the Neisseria meningitidis strains are sensitive to PBT2. They all have very low MIC. And here is the list of the mode of actions of PBT2 that we discovered and also we proposed. And then the first one here, disrupting metal homeostasis, most likely to be the Achilles heels to Neisseria gonorrhoeae. So by using this one, and we look at the structures of a PBT2, and then the core structure of PBT2 is actually this 8-hydroxyquinoline. And using this structure, we then synthesize and develop different classes of iron 4. And we call them new chemical entities. And due to the limits of time and also the confidentiality, I can only quickly summarize what we have known, and then we for this new chemical entity, we have a few lead compounds that have shown that they are more potent than PBT2 and less toxic than PBT2. They show bacterial cytoactivity and no antibiotic, no resistance observed after 30 days of treatment. And our proteomics analysis also indicate that all these new chemical entity modulate a certain key pathway with a high efficacy. And we are very confident that all these data support our future development of novel antibiotics to treat multi-drug resistant Neisseria gonorrhoeae. So in summary, the 3 key points, and the first one would be the most important take-home message would be multi-drug resistant Neisseria gonorrhoeae is a serious antimicrobial resistance threat and an escalating public health concern. And we're very excited about the PBT2. It is a repurposed drug originally developed for neurodegenerative disease, but it has shown the promises of novel treatment for Neisseria gonorrhoeae and possibly other pathogens, including Neisseria meningitidis. And then the third one is we're very excited about building on our understanding of PBT2 structure, mode of action, and we know about the pathogenesis of Neisseria gonorrhoeae. We have developed the whole range of new class of novel chemical entities which will enhance antimicrobial activity, and they potentially superior to PBT2. And finally, I would like to thank to all the people in this antimicrobial resistance drug development, people involved in these projects, and especially Professor Mike Jennings and Professor Mark von Einstein. Mike is very expertise a microbiologist in pathogenic Neisseria as well, and Mark is a biomedicine chemist, and all the chemists and all the room in the mass spec analysis, and also Jennifer in the ex vivo gonococcus infection model. And thank you.
Thank you, Freda. That's really exciting work. So our next speaker is Anthony Coates. Anthony is the founder and chief scientific officer of Helperby Therapeutics, a biopharmaceutical company dedicated to developing the next generation of life-saving antibiotics. The company has several therapies and clinical trials targeting unmet needs in areas of significant market value, and including candidates for urinary tract and skin infections. Anthony is also professor of medical microbiology at St. George's University of London. He leads several research teams, is the author of over 180 publications, and has edited 13 books. He is an inventor on 200 patent applications, and of which 127 have been granted, and was a member of GARDP's Scientific Advisory Committee until 2025. So welcome, Anthony. Please start your presentation.
Well, thank you very much, Jennifer, and hello to everybody. I'd particularly like to thank GARDP for organising this webinar and for inviting me, and of course to Victor for helping me with this slideshow. So I'm going to talk about the development of zidovudin as a repurposed antimicrobial. And my key messages are that bacterial resistance, of course, as we all know, is increasing. This is leading to the destruction of antibiotics, and this is leading to an increase in sepsis deaths from antimicrobial resistance. As Jennifer has said, the pharma industry is not generating enough new antibiotics, and also repurposing the antiviral zidovudin as a low-cost antimicrobial in combination with colistin or meropenem leads to a synergistic kill of highly resistant bacteria. So those are the main messages that I want to convey. So I'm going to start with a short story about Carl. Carl is a nearly 3-year-old, and this story is told by Catherine, his mother, and is shortened by me. So one day Carl developed a cough, and the following day he developed stridor, which is a high-pitched sound due to obstruction of the upper airways. The GP started Carl on steroids and sent him home, and the next day Carl had a low fever, but the following day his fever was very much higher, his breathing was difficult, and he was lethargic. The doctor did blood tests which showed an infection, so he was taken to ER casualty. He was diagnosed with respiratory syncytial virus pneumonia and asthma. He spent 11 hours in ER. He was then admitted to pediatric intensive care and was diagnosed later as suffering from the bacteria MRSA sepsis. He was put on antibiotics. He was treated with high pressure air, had a tube down his throat to deliver oxygen to his lungs, and many other procedures. Unfortunately, his sepsis led to shutdown of the major organs in his body and also to his legs. He spent a total of 106 days in the ICU, and after amputations and skin graft surgery he went home, and after a further year of recuperation, I'm delighted to say he's able to walk, run, climb and play with his peers today. His mum says, "Hug your babies and be grateful for every day." So as we know, there has been it's an unexpectedly high growth in resistance, especially into low to middle income countries. And this is between, along the bottom axis, 2014 to 2018, a 10 times actual growth in 4 years. And in the US, a 3.6 actual growth in 4 years. And there's also been an unexpected high growth in global sepsis deaths from antibiotic-resistant bacteria. And over the last, this projected deaths is up the vertical axis, and date in years is along the horizontal axis. And what this shows, actually in orange, is the predictions of deaths from all antimicrobial resistances, from all organisms, by Lord O'Neill and his UK commissioned report in 2016. And the red dots show a doubling actually over the 5 years between 2015 and 2019. And that is all is known at the moment. But of course, we hope that that won't carry on for bacterial sepsis, because otherwise it would mean a much earlier reaching the 10 million dead per year, which Lord O'Neill identified. So now we need new antibiotics, and I will tell you the story of Jen. Some of you know it. They developed a new chemical entity called plazomicin, which is a derivative of gentamicin and then sizamicin. And Jen raised about $600 million US dollars, including 96 million from the US government, did several phase 3 clinical trials, and entered the market with FDA approval in 2018. Their highest valuation was $1 billion, and they entered the market with a sales price for a course of antibiotics of $10,000. But unfortunately, they couldn't sell enough at that price and went bankrupt in 2019, like so many others, other small companies making antibiotics. And plazomicin was sold to Cipla for $16 million under Chapter 11 in Florida. So what can we learn from the Jen bankruptcy? Well, I think first we need to accept that new chemical entity antibiotics are not commercially viable. And I'd like to point out GARDP's wonderful work, an extremely important role in addressing this issue. And they are certainly the world leaders in this field. So the development costs are too high, and actually most big pharma gave up years ago, and almost all small pharma fail in the market, like Jen, at Paratech, for example, and so on. We need to develop a lower cost way of making new effective antibiotic treatments. And I would say they need to be nearer the generic price, which is the price of the generic companies when they take on manufacturing and selling patented drugs when the patents have run out. And in particular, I believe that we should try to repurpose old drugs in synergistic combinations. And Freda has been talking about synergy earlier. So what do we need? Well, we need a commercially viable antibiotic company to start with. And my company, Helperby Therapeutics, is potentially commercially viable because it has a low-cost platform technology. And this low-cost platform technology is repurposing of combinations of low-cost synergistic drugs. This is about 10 to 50 times lower cost than making a new chemical entity. So what is synergy? Well, I'll just repeat that it's antibiotics which work together to produce an effect more potent than if each antibiotic were applied singly. So additive is just 1 + 1 = 2, but synergy is 1 + 1 equals whatever, 100 or more. And I'm going to talk about azidothiamidine, which is also called zidovudine, and it was it's first developed as a anti-cancer drug, but it was then used to prevent and treat HIV/AIDS. It was the first anti-AIDS drug, anti-HIV drug, which was developed. But it was also known from 1987 that it was active against gram-negative bacteria. So why develop zidovudine as a repurposed antimicrobial? Well, it's low cost, low toxicity in humans. It's synergistic with colistin and other drugs in combination. And these combinations can kill highly resistant bacteria. And colistin can be used at a lower concentration. We've shown about 1/3 the dose. And this, again, lead to reduced costs and lower side effects as far as colistin is concerned. So this shows published data. AZT is azidothiamidine in combination with colistin produces a synergistic bactericidal activity against, that's carbapenem, that's NDM, metallo-beta-lactamase producing Klebsiella pneumoniae. And up the vertical axis, log colony forming units and time in hours along the bottom line. And you can see the controls at the top, the control of colistin and the control itself and AZT on its own. But when you put the two together, you get this red line, which kills 10 to the 7 organisms, 10 million organisms per ml. So the next slide shows azidothiamidine and colistin are synergistically or additively active against 100 colistin and carbapenem resistant Klebsiella pneumoniae isolates. And on the left-hand side, you can see the synergy, which is measured by one of the classical ways of measuring synergy, which is the fractional inhibitory concentration of less than 0.5. 79% were synergistic and 21 were additive. And it's also active in animals, in mice, log colony forming units up the vertical axis, hours of treatment along the bottom axis, controls, the 3 lines at the top, and the red one is AZT plus colistin. This has been done in a number of different animal models, actually. So we've also done a phase 1 clinical trial in 27 volunteers, and we did all the classical things that we need to do, which is we looked at safety, of course, and it was safe in its combination of azidothiamidine and colistin. We also did, which I've not shown in detail here, but we also did ex vivo studies in colistin-resistant E. coli, and we showed that levels of drugs which were able to show in humans in our phase 1 trial, when they were put in test tubes with colistin-resistant E. coli, they killed them very nicely and synergistically. That's also been published. And AZT synergizes with meropenem and actually other carbapenems against NDM or carbapenem producing Enterobacteriaceae, and these also show the same thing but with different concentrations. So the top 3 lines are the controls, and then the sort of line almost going down very steeply is the combination. Synergistic. So progress so far for Helperby Therapeutics Group Limited is 130 granted patents worldwide, 150 million market cap, and a contract signed for 75 million from the USA. So I want to finally tell you a low-cost story about Kenneth. And Kenneth Giles Friedkin was born in Kansas in North America. And his father was a Russian-born Jewish tailor, and his mother was American. At the age of 8, Kenneth saw an air show in San Diego, and he was absolutely fascinated. He obtained his pilot's license at the age of 17. He owned a flight school, not very successful. He tried aerial fish delivery, banner towing, and a charter airline. I'm afraid had no success with any of those. Then in 1949, he founded a low-cost airline called Pacific Southwest Airlines. He bought a Marine Corps latrine, which served as the carrier's first ticket office. He had no check-in area and had to weigh passenger luggage on his own bathroom scales. This was the first low-cost airline. In 1986, it was sold for $400 million to US Airways. So what does this tell us? Well, low-cost products such as air travel change the world we live in. And one day, perhaps low-cost, effective, repurposed antibiotic combinations may also change the world. So in conclusion, new chemical entity antibiotics are not commercially viable. The costs are too high. Low-cost zidovidin plus colistin kills highly resistant bacteria at a low dose of colistin. And I'd like to acknowledge in particular at St. George's Dr. Yanmin Hu, and she and I are inventors on all these patents, and the Helperby Therapeutics Group, particularly James Phipson, the CEO, and grants from the EU, MRC, BBSRC, British Business Bank, and Helperby Therapeutics Group. Thank you.
Thank you, Anthony. That was very interesting and exciting new direction. So our next speaker is François Franceschi. François is an associate director of the series Bacterial Infections at GARDP. He has over 30 years of experience in the area of antimicrobials, previously serving as program officer for therapeutics development for antibacterials and antifungals at the NIAID in Bethesda, Maryland. He has also held various director positions in antibiotics, including R&D at RibX, and was the principal investigator at the Max Planck Institute for Molecular Genetics in Berlin, Germany. At the Max Planck Institute, his research was devoted to the structure and function of ribosomes, especially when they're in complex with antibiotics. His group was a pivotal part of the international consortium led by Ada Yonath, who won the Nobel Prize in Chemistry in 2009. François received his PhD in chemistry at the Freie Universität in Berlin, Germany. So welcome, François. Please start your presentation now.
Sorry, Francois, you seem to still be on mute.
Can you hear me now?
Yep.
Okay, sorry, I don't know what happened here, but yeah, I say everybody welcome. This is a One Health approach to AMR. Is GARDP's exploration of a veterinary antibiotic for its therapeutic potential in humans. And this antibiotic is called apramycin. Some of you have sent me some message saying, is that really repurposing? It's not a different therapeutic area, but I mean, it's repurposing in the sense that this is an antibiotic that has only been used in animals, and we are trying to explore its potential for use in humans. I don't know why this is not moving. Okay, there we are. So for those of you that don't know about GARDP, so our vision is that all infections are treatable for everyone, everywhere. And our mission is to accelerate the development and access of treatments for drug-resistant infections. And then the area where we focus is in the intersection between priority diseases and infections, key regions and populations, and the WHO priority pathogens. Because as you know, a lot of the heavy burden of pathogens are mostly localized in low and middle income countries. So just to, I don't know what's happening. Okay, there we are again. It's taking a while, sorry. Okay, apramycin is a veterinary aminoglycoside antibiotic. For those, I give a very short introduction about aminoglycosides. They have been a cornerstone of antibacterial chemotherapy. Streptomycin was the first one isolated from Streptomyces griseus and introduced into clinical use in 1944. They are all very potent. They are bactericidal. That means they kill the bacteria, not just arrest their growth. They are broad spectrum, and they act by inhibiting protein synthesis in bacteria. On the golden age of antibiotics, let's say, so following streptomycin, we had neomycin, kanamycin, gentamicin, tobramycin, amikacin. Some of them are derivatives from some of the original ones. Arbicacin, and then the last one that was developed and that Anthony touched in his talk was plasmomycin in 2006. That was a derivative from cizomycin. Although some of them are still widely used in the clinic, a shift away from systemic use of aminoglycosides began in the 1980s, because at that time, then third generation cephalosporins, carbapenem, and fluoroquinolones came into the market, and they were perceived to be less toxic. One of the toxicities associated with aminoglycosides is either ototoxicity or nephrotoxicity, and they also were perceived to have broader coverage than the aminoglycosides. Of course, then this shift led to apramycin, because that was discovered in the 80s or developed in the 80s, to being only approved for veterinary use. Here again, you can see more or less the development. Apramycin, as I said before, was only approved for veterinary use, but there were some very critical difference between apramycin and all the other aminoglycosides that are currently used in the clinic. For once, apramycin was monosubstituted, and second, it was a bicyclic deoxystreptamine, as opposed to all the other aminoglycosides that are reversed. They are disubstituted and monocyclic. Apramycin was shown to have a very broad spectrum of activity. Importantly, apramycin also seems to be resilient to almost all aminoglycoside resistance mechanisms typically found in multi-drug resistant and extremely drug resistant gram-negative bacteria, including the 16S RNA methylase that is very common in carbapenem resistant and metallo-beta-lactamase producing strains. In the slide, you can see in the bottom left, this is just showing how the aminoglycosides bind to the ribosome, which is the target for protein synthesis, and showing that this methylation, that is the cause of resistance for a lot of those strains, does not affect apramycin because of its unique structure. There is only one clinically relevant resistant mechanism via ACC3-4 that confers resistance through acetylation. But that has been shown to be very low prevalence in clinical isolates, and we will talk a little bit more about that later. And again, preclinical and animal data suggests that there is, importantly, that for apramycin, it seems to be that this aminoglycoside has lower nephrotoxicity and lower ototoxicity than other aminoglycosides. So the WHO pathogen priority list from 2024, the two most relevant groups are the critical group and the high group. And based on published information and information that has been generated by Juvavis, the company who was trying to develop apramycin, essentially apramycin has activity in vitro and for some of them also in animal models against all the pathogens, meaning critical group and high group, even including Mycobacterium tuberculosis, which was recently published in 2025. A little bit of the development history of apramycin. So as I mentioned before, it was authorized for use in animals in the EU in 1980. It followed then in the USA. Then in 2015, a small biotech was funded in Zurich, a spin-off of the University of Zurich, to support the development of apramycin, meaning its clinical development. They got substantial funding from the ENABLE program from the EU, and there was essentially apramycin was the only drug from that program that advanced to the clinic and went into phase 1, at least on the first iteration of the ENABLE program. They did carry out a phase 1 study first in human, and they showed that they could reach a predicted efficacious dose, and that there was, at least in this very small population where it was tried, no significant or no adverse events. They had planned then to run a phase 1b, which was a multi-dose, a multi-day dosing of the antibiotic in patients with complicated urinary tract infections, to look for safety and also to have a very initial idea of efficacy. This trial was ready to go in 2020, but it never started because that was at the time when COVID struck, so there was not possible to run those clinical trials. And by the time the COVID wave was gone, the funding had expired and the trial was never run. In the meantime, one of the things that was very important to know was, is this antibiotic getting into lungs? Because if you want to combat, for example, hospital-acquired pneumonias or ventilator-acquired pneumonias, you need to know if the antibiotic, that particular antibiotic gets into lung. Here, NIAID, the National Institute of Allergy and Infectious Disease, came to the rescue. They decided we are going to perform a phase 1 to see if the antibiotic gets into lung, because some aminoglycosides do not penetrate well in lungs. This trial was concluded last year. The clinical study report came out this year, and it does shows, importantly, that it does get into lungs into reasonable levels. Sadly, in 2024, as this trial was being completed, because of some of the reasons that Anthony was speaking about and the whole antibiotic, say, market and funding situation, Juvavis went into bankruptcy, and the IP of apramycin went back to the University of Zurich. GARDP, thinking about all the money that had been invested in this compound and the potential that it has, decided to try to rescue that. And then in June 2025, so just a month ago, we got the data and the rights for apramycin and the IP from the University of Zurich. And so we decided to start trying to rescue this compound because of all the potential we see that it has, especially for treating infections in low and middle income countries. One of the first questions we were faced with was the people were saying, well, this is a veterinary drug, so what about the veterinary use and what they are going to say? So for both the WHO and the World Health and the World Organization for Animal Health, aminoglycosides are listed as veterinary critically important antimicrobial agents. In the case of WOAH and for WHO, it's a critically important antimicrobial for human health. In the next slide, we can see that how is this classification, how this prioritization of antimicrobial classes in the WHO medical important antimicrobial list. We can see that plasmomycin is the only aminoglycoside that is in the authorized for use in humans only, and that all other aminoglycosides and macrolides, for example, are in the critically important antibiotic category that are sometimes used in animals as well as in humans. As I mentioned before, we also had a conversation with the World Organization for Animal Health, where we discuss our intentions to get into apramycin and try to determine its potential for treatment for human pathogens. And they agreed with us that it was important to explore that, and they didn't see a risk that this was going to be a problem also on the veterinary medicine. Even so, apramycin is not widely used, it's still used in veterinary medicine. Okay, there we are. So then a question also that we were often confronted with is, well, apramycin has been, used for 40 years in veterinary medicine. Is there already resistance in human isolates? And after 40 years of veterinary use, the data at this point at least shows that there is little reason for concern. There was a study about like almost 600,000 genomes of pathogen gram-negative bacteria deposited at a database. And then since there is one predominant and the only one significant in the clinic mechanism of resistance for apramycin, they went and looked for that gene in that whole genome database, and they detected that only 0.7% of the isolates were having that particular apramycin resistant gene, despite of the almost 45 now years of veterinary use. Then also Juvavis and their collaborators, they published, and also independent researchers across the world, they have published several studies where they show that, for example, carbapenem-resistant hypervirulent Klebsiella strains from China were all sensitive to apramycin. Also from 470 multi-drug-resistant gram-negative bacteria isolates from Cambodia, Laos, Singapore, Thailand, and Vietnam, 98.3% were susceptible to apramycin. And so even apramycin-resistant Acinetobacter baumannii and Pseudomonas aeruginosa, only 2% had an MIC that was greater than what may be able to be covered with this drug. So there have been even a speculation that apramycin, that the lack of apramycin resistance may be linked to the fact that if you acquire apramycin resistant, maybe this has a fitness cost, and it was a publication in Nature in 2025 alluding to that. So this also, again, shows the same. Essentially, this is a distribution of minimal inhibitory concentrations. Blue is apramycin, green is amikacin, which is one widely used aminoglycoside in the clinic. And you see that all the minimal inhibitory concentrations for apramycin are towards the left side, while you have very, very high resistance to gentamicin and amikacin, just solidifying the fact that definitely apramycin seems to be better than amikacin, and we plan to redo the studies with some new strains in the very near future to support the case. This is just to show that there have been a lot of different publications in the last, say, 5-6 years, showing the potential of apramycin for treating very high resistant bacteria, and also pointing out to the fact that in animal studies and in vitro studies seems to have lower autotoxicity, which is one of the main issues with aminoglycosides than all the other aminoglycosides used currently in the clinic. So what are we planning to do? So now that we have the rights, we have decided that, I mean, there are a couple of things that we need to do in very short term or medium term. We are going to generate some pharmacokinetic and pharmacodynamics data in animal models and those hollow fiber infection models to investigate, the current dose that is being used in the clinic, and also to investigate synergy among apramycin and other compounds. Because as Anthony was pointing out, some of these older compounds, when you mix them with, for example, like carbapenem, they tend to be synergistic and have a much broader spectrum, and then lower the potential for generating resistance. We also need to generate material from veterinary supplies, at least to start or restart the phase 1B that never was carried out. That already is a protocol, a clinical study protocol that had been approved, and every approval had been done. So we are going to reestablish contact with this group to see if that trial can be restarted in the short term, this phase 1B trial that we recruit about 40 patients. We definitely need to also establish a supply chain for apramycin, find out if there is facilities in the EU maybe or in India that can be used to produce apramycin. And importantly, one of the things about apramycin, and also going back to what Anthony was saying, is this is a fermentation process, meaning you essentially have to grow the bacteria that produces apramycin, which is called Streptomyces tenebrius, in a big tank of, say, 1,000 or 10,000 liters. And from there, you isolate the antibiotics. So there is no synthetic steps, no nothing. It's just a purification out of culture of growth media, meaning this has the potential to be a very cost-effective antibiotic for use in LMICs. And of course, we are also already starting to plan what could be the interactions with regulatory authorities to define what is the regulatory path, how are we going to start developing investigational plans for pediatrics and so on, because we think this compound could also be a very important addition to the treatment of neonatal sepsis, especially in low and medium income countries. And then to finish, why apramycin? Well, it's a known class of antibiotics. It has known liabilities, but it has the potential to cover all the critical priority pathogens according to WHO. Based on the known synergies between aminoglycosides and beta-lactam antibiotics, apramycin has the potential to become a solid backbone for combination therapies, for example, as neonatal sepsis, as I mentioned before, and also for serious bacterial infections in adult populations. It appears to be less ototoxic and less nephrotoxic than the currently used aminoglycoside. That could be a big plus because at least the animal model shows that it doesn't accumulate in renal cells, and it also doesn't accumulate in the cochlear hair cells compared to the other aminoglycosides. So bottom line, it combines premium coverage with excellent cost of goods. So meaning that potentially this will be a few dollars per dose. And it also would allow GARDP to explore a new path for the development of a low-cost, high public impact antibiotic that could be easily deployable in low and medium income countries, and of course also in high income countries. The next slide is just thank you to all our funders, and thank you to all the R&D team at GARDP, which without this wouldn't have been possible. Thank you so much, everybody.
Thank you, Francois. That was great. So at this point, we're going to switch over and start the Q&A session. As a quick reminder for the audience, you can submit questions as shown in this slide, and we'll do our best to respond to as many questions as possible within our time frame. So I think if all of our speakers could turn back on their cameras. I think Freda, maybe we'll start with you. We have a couple people have asked similar questions. Have you looked at PBT2 or some of the other NCEs, if they would have activity against other STI pathogens, particularly like chlamydia or mycoplasma?
Yes, we have a plan to do that, but we are still in progress, because that would be another sort of ultimate goal. You have one drug for both, especially chlamydia is the #1 in STI.
Be very interesting to see if there's activity against other pathogens. Yeah. And then maybe kind of a similar question here is several approved drugs are based on the 8-hydroxyquinolone scaffold and are indicated for diverse medical conditions. Have you screened like a library of other approved drugs to explore their use?
No, we have not. We only look at especially the small molecule, which will be the PBT2, because it's quite small. And that is what we focus. And I did look at other INO4 using agriculture, to be honest. And then also, then we don't actually look at other one. And then the reason we want to stick with the PBT2 is we know it's safe for human use. And then even for repurposing, that will actually, like everyone, it will save a lot of cost for us to move forward.
Excellent, thank you. I think a question for Anthony. If a low cost, I think this could go to multiple speakers, but if a low cost is the desired outcome, we also need to find a way to develop, reduce the cost of development. How can we get regulators and drug development processes on board with this, kind of changing the regulatory pathway to make it more financially feasible for antibiotics?
Yes, well, there are a number of answers to that question. I mean, the first thing is to start with a repurposed drug or repurposed to repurposed drugs. I mean, it's simple stuff, really, that repurposed drugs are manufactured. If you choose them carefully, like François has done, then you can find places around the world, particularly in LMICs, which will be manufacturing these drugs at very low cost. But they need to be drugs which have, if I might say so, been around, because otherwise, if they're new chemical entities and they just don't seem to penetrate into LMICs very well, and therefore you don't have the manufacturers. And the manufacturing is extremely important. And I know GARDP is concentrating on that very much, which is exactly the right thing to do. So I think the first thing is you really do need to have widely available, particularly LMICs, manufacturing of low-cost drugs. The second thing is the pathway, the regulatory pathway for these sorts of drugs can include things like the 5052B pathway, and they can include a phase 1 trial, no phase 2 trial, and only one phase 3 trial. Now that contrasts with some drugs which have to have phase 1, phase 2, probably several phase 2s, and certainly several phase 3s. And that is much more expensive. The other problem, of course, with new chemical entities is the unexpected. And the unexpected, I'm not going to go into any details, but François and I, over the last few years, have seen a few unexpected things which happen to new chemical entities. And it can kill the new chemical entity, or it can be just very expensive to actually address the issues which come up. So I think that the reasons are not just low cost manufacturing. The reasons are also a lower regulatory pathways, and the regulators we have found are more open to this sort of thing now than they certainly were 10 years ago.
Do you think with these drugs that have already been developed for other disease states, is it possible to reduce the safety studies or some of the toxicity studies that have to be done by using the previously generated data, or do you think maybe that data is too old or doesn't meet current regulations?
No, you can certainly use those data, which is another reason that you can lower the costs, and the safety studies will be pretty dramatically reduced, really for two reasons. First of all, that many of them been done before, and secondly, they've been in humans for 20, 30, 40, 50 years. And so the experience in humans is very widespread. If you use a drug that's been used widely, the experience is used widely. And of course, that gives doctors more confidence in using a combination which contains, say, two drugs which they've used before.
I think, and Freda, have you seen this as you've talked to other, physicians in the field, that they're open to like repurposing these like antivirals or other sort of, other drugs to be used in the anti-infective antibiotic kind of space?
No, I have not, but we are looking because we are working with the STI clinics and then planning to do some clinical trial work that we can do. But that we're still planning to do that. And about what you say, if they're willing, for example, if we have talked to the STI doctor about if they're doing, I think for gonococcus treatments, they're already, due to the gonococcus so resistant to so many antibiotics, and they are already doing synergies. They're doing multiple antibiotics administrations to kill the disease actually. So actually, me personally, I have not actually talked to the physicians about this, but then we're more in the research sort of work that we will we're talking to them about doing clinical trial, what we think that will work.
Thank you. I think there's quite a few questions coming in, so thank you, everyone. So here's for Francois. Have you thought of human behavior in the irrational use of apomycin? For the regulatory authorities for LMICs, have you considered like illegal pathways for antimicrobial use in these countries? I think there's another question that's come in also about as you develop these compounds that have already been used, how do you navigate IP challenges and use in these countries? Maybe Francois, you can speak to that.
Yeah, First, I want to compliment a little bit what Anthony was saying about the development path for the repurposed drugs. I totally agree with Anthony that repurposing a drug has a lot of advantages because it's a known entity. You can go from phase 1 to phase 3, etc. But I also want to add that in some cases where you probably see, okay, there is a need to develop a new chemical entity, there is ways to do that, and I would say GARDP's way is also like trying to look from the very beginning, like, will this drug be possible to will it be possible to manufacture this drug at a reasonable cost? And that's one of the things that GARDP does from the very beginning. The two drugs that we currently have in the portfolio that are being manufactured by GARDP, soliflodacin, which is a gonorrhea drug, and cefidrochol, the idea was to find manufacturers in LMICs that could significantly reduce the cost of production. And I guess if you go with that mentality from the very beginning, I am not saying, and I agree with Anthony, that developing a new chemical entity has a lot more challenges than a repurposing. But looking at that from that way, at least increases your chances of success because you are having those thoughts from the very beginning. About IP protection, yes, that's definitely a challenge in the sense that, okay, the University of Zurich has some IP on apramycin. It's not going to last forever. Say it's going to probably expire in 2030. Maybe that drug will already be by then approved. There are some mechanisms, like for example, if you get QIDP designation, or in the EU, you get to like 5 years exclusivity. So there is some incentives there. There could be also ways of, for example, improving the manufacturing of apramycin in the sense of modifying the strain that produces to get more purity and then generate some IP for that. That's not so important for GARDP. It's more important to find a partner that then will see, a commercial viability of the project there, and that's something we definitely keep in mind. But yes, animal use, as you said, if apramycin evolves as a drug that could be used in human, probably it will be looked for with scrutiny on the veterinary side to say, for example, this shouldn't be used for, say, growth promotion or prophylactic, only for treating, animals. Even in animals, the resistance seems to come and go. So that's also kind of puzzling, so that it's not a lot of resistance out there. Also, the way it's used in animals is completely different. It's not systemic. We are looking for apramycin to be used as an IV infusion. In animals, it's used just orally. It's pure absorption. And those type of drugs are not normally absorbed into the bloodstream. It's more about, combating gastrointestinal pathogens in that case. So yeah, these are all things that we have in mind, and we are trying to look at initially. Yeah.
Jennifer, could I just add something?
Please.
I just wanted to say how much I very much agree with what Francois is saying and support what he said. Regarding our patents, Helperby Therapeutics, what we have found is that, first of all, this question comes up often. How can you possibly patent 2 old drugs? And the answer actually appears in all of our patents to be the synergy. And the synergy is often unexpected. Well, it is. It has to be unexpected. And the synergy is the thing that patent officers find convincing, and that really what underpins virtually all of our patents, not all of them, but virtually all of them.
So I was thinking when you were discussing, I'm going to mess up the name, but zidovudine. Thank you. Are you planning? Do you foresee this to be patented and commercialized as a combination drug, or as a standalone drug that could then be added to current therapies?
No, it's a combination drug. No, we're not suggesting it is used on its own.
Yeah.
As François knows, I'm a great fan of combinations because yes, there are some difficulties with developing combinations, but I think the potential for highly resistant bacteria is probably going to be combinations in the future. And this is why we're developing combinations, and indeed others like Freda and Francois are developing combinations.
Okay, I think maybe we can go back. Well, here's a general question as well that I think all of you could weigh in on. But is it possible to predict AMR to new antibiotics using AI tools? Or I think similarly using AI to look at new compounds? Any thoughts there?
It's a tough one. I mean, as we know, bacteria have been evolving for millions of billions of years, so they always find a way. And that's why I think what Anthony is also saying, combinations are a very important tool, because then you are hitting the bacteria with two different hammers, put it that way. And then it's more difficult to then develop resistance when you're hitting twice. Than, I mean, in some cases it has been shown, in some cases the jury is still out there, but clearly combinations are an important tool for trying to avoid that. I mean, resistance is always going to emerge. The bacteria will always find a way. And that's why we need to have a robust pipeline, because, I mean, unless we find a vaccine in the near time, in the meantime, that, will combat or prevent all those type of infections, which at this point doesn't look very promising, the bacteria are always going to outsmart us and come up with resistance. So that's why we need a very robust pipeline, and the use of combinations is a very important, and also the whole understanding of pharmacokinetics and pharmacodynamics, because, I mean, if you underdose a drug, then of course you are going to have more resistance than if you don't. And this field has been evolving a lot now, and all the regulators also put a lot more importance into understanding pharmacokinetics, pharmacodynamics. What is the proper dose, so that I don't develop resistance, right away for a particular drug. But I let Anthony and Freda to say more.
So I think,
sorry, Freda.
sorry.
No, you go,
okay.
Freda.
Okay, no, I was just thinking that to predict this most of the time, in my point of view as a molecular biologist, I feel like to predict this most of the time is a mode of actions of the drugs. And of course, when it's synergistic, that you have two, of course, like Anthony mentioned previously, 1 + 1 = 100, and then that is become very big. But maybe the bacteria takes a longer time to develop a resistance. And how can AI predict that? If we know mode of actions, and those things probably can be predicted. And for example, I understand that the development of a gonorrhoeae antibacterial, there's a new antibacterial drugs that is going, like, it's under the, I think it's in phase 3 clinical trials now, and then they know what proteins that the drugs will target to. So a lot of researchers and also a lot of people, they're doing study that if these mutations in these things would cause the resistance to the drugs. So I think to answer that question, can I help? I think if we know the target proteins and then we know, I think the AI will be able to help with that.
Yes, if I could just say that I agree with you, Freda, that I think that AI will help predicting AMR, for example, perhaps, and maybe even new drugs. But the prediction of AMR, I think we also have to take into account what François has said, that it's probably inevitable, even to combinations, that bugs are going to become resistant. It's just as they become resistant to absolutely everything. And so what I think we probably do need is platform technologies which can yield multiple different drugs for the future. I'm not talking about tomorrow. I'm talking about in 10 years' time, 20 years' time, 30 years' time. And rather like Freda has been developing new chemical entities, but multiple ones. I think that is the way forward. And I haven't talked about it this morning, but our platform technology has actually already identified 5 different treatments for highly resistant, carbapenem resistant WHO and CDC pathogens. But there are many more, because if you think about a tumor, that's Zardovin and say meropenem. Hey, what about a threma? And then what about a former? And what about a 5-mer? And all of this, people sort of raise their eyes and say, oh, don't be silly. But actually, if you want a platform technology to last for say a generation even, you have to have more than one drug. And it's much more preferable, I'd have thought, to have a platform technology like perhaps Freda's, which can produce lots of different drugs, or our platform technology, which can produce lots of different drugs over the years. I'm not thinking about my lifetime. I'm thinking about children's lifetimes. That's what's needed.
Definitely, I agree. There was a more general question kind of about how you go about with lead identification process for identifying other drugs to be repurposed. Is this like a high throughput, panel you're testing? How are you kind of identifying these different leads from a high level view? Are you using AI? I think maybe not yet, but maybe this is what's going in the future.
Well, I mean, AI would be one way of doing it, but it's sort of unproven yet. So I think either you have to have a robot type of screening, or you have to have a robot plus AI screening. I would have thought that would perhaps be one answer for the time being. I don't know what my other panelists feel.
Or I think what I know about what we do about drug screening, again, if we know, for example, you know the bacteria well, and then you find the important functions of the protein, and you know that loss of function of that protein will cause a lethal phenotype. A lot of people would, firstly, computational. They have lots of compound library or the repurposed or the drugs library. They can screen, use a computer to screen if any of these drugs can block the function of this protein. And then once that, they pick a good one and then start doing screening, study in vitro and then in vivo and then etc. So that is some research that do these kind of things. Or another thing is, for example, we have a facility of Compound Australia. They already have like a library of compounds stored in there. So what researcher can do is, so for the microbiology, they can screen by MIC, and then we also have things, and to see for those one, people screen it. If they found something that can kill, most of the time, they will need to find what's the mode of action. The same story as we do to BTG. What is the mode of action? After they find the mode of action, the same thing, they can look at the structure and start modeling how it affects it. I also know that some people here use things like SPR to screen lots of compound and then to find if the target protein, if they know the proteins, the functional protein, how they bind any, like they can screen lots of drug, like the known drugs there, and to see which one can bind and has the high affinities, and then they can use that to test, to do the in vitro and then in vitro test, and then have generating data, and then to see if they can use in animals to do in vivo test. So this is what I know in terms of high throughput screenings of the compounds.
Thank you. So we just have a little bit of time left. I think there was a question here about challenges with susceptibility testing when you're using combinations, or how do you choose the combination drug that you want to use? Often it's colistin. Have you looked at other combinations? I think Anthony, Freda, Francois, any thoughts on that?
I have an issue with colistin. I'm sorry, Anthony, you can probably answer. When we first time did this, it's more a discussion. When we first time send our study to review, and a review jump up and then even refuse to review because they say polymyxin and colistins are not safe antibiotics you should not use that and you should not study it. So, and that's why we start looking into any other combinations, and that's why we moved the study to the Nature cation peptide LL37 and PP1, and then we found that the PBT alone will be enough to sensitize the repurposed bacteria. So I don't know, Anthony, you can answer that. It's my question. I've been thinking along while the presentation as well. But about Jennifer, what you said about this, right? I think a lot of time we try to the antibiotics we want to use is the clinical relevant antibiotics first to see if our repurposing drug will work, and then that's what we do in the laboratory, like the first thing we do.
Yeah, well, Freda, I get the same criticism about listing often, and it's fine, you know, people can say what they like, but then what are they going to use? And the answer is they're not going to use anything except for colistin, because that's the only thing that's left in many low to middle income countries. Can you make colistin safer? Well, maybe you and I can make a combination which will yield a lower dose of colistin. And heparin was the classic example. Many years ago, heparin was a dangerous drug, and everyone knew it was a dangerous drug. And then they introduced low-dose heparin, and it just filled the entire world and was a huge success. So I think low-dose colistin is probably the way forward for colistin, or change it to something else that people would accept, like meropenem, for example, or one of the carbapenems, and make your combination synergistic with using a carbapenem instead of colistin for those that really get indigestion, serious indigestion from colistin.
All right, I think that's really exciting. I think we'll see how it all goes. And GARDP as well is looking at different combinations and how to develop there. So thank you all. This was really a great discussion and great presentations today. I'd like to thank everybody for their questions and comments, and thank you for our speakers. And so now, Victor, I'll hand it back over to you.
Great. Thank you very much, Jennifer, for moderating today. And of course, thank you very much, Freda, Anthony, and Francois, for your excellent presentations. So you can already register for our next Revive webinar. This will be on overcoming the challenges of drug discovery and development for tuberculosis, and will feature presentations from Jeremy Rock, Dirk Schnappenker, and Ola Kakon. And this will be on the 9th of September. So that will be all from us. Thank you everyone for joining today and for contributing to this discussion. I hope you found the webinar interesting and useful, and that you'll join us in the future. And please do spread the word amongst your colleagues. And please also fill in the survey afterwards to inform us for future webinars. Thanks again, and goodbye.